maybe one of the last data sets that we should talk about from ESMO is a signal of where the puck might be going, and that is, how do we start building on the backbone of EV-Pembro, and could we start combining some of these newer drugs like the antibody-drug conjugate? So there was a phase 1 study that looked at the combination of sacituzumab, govitecan, another drug that has an approval in this disease, but may be struggling to find a place. So SASI plus EV, and I don't know if you guys saw that data set. It had a really high objective response rate in a refractory population. So maybe Jonathan, I'll start with you. Do you think this idea of combining ADCs will have a role?
I think it will, in fact. And we've known in oncology forever that single drug therapy doesn't have the maximal benefit, that you need to combine treatments with different targets, different toxicities to get the maximal benefit. And so I think the big question from this is going to be what plays well with the third agent would be immune checkpoint inhibitor, and whether the two-drug combination plus a checkpoint inhibitor is going to be better than EV-Pembro. This two-drug combination plus Pembro is going to be better than EV-Pembro, to me, is unclear. I think it's clearly worth exploring, and there are a lot of other ADCs that are out there now, and with other cytotoxics, other TROP2 ADCs that are investigational. And I think there's some real interesting opportunities for combinatorial therapies, and this is maybe one of the first times that urothelial cancer is actually leading the way. This was the first double ADC combination trial, I think, that's ever been reported. And I think we should see more of these in bladder cancer going forward.
Matt, do you want to comment on that?
Yeah, I think it's interesting data. I mean, the principles of independent drug action, right? One of the principles is that giving drugs together rather than sequentially could be beneficial if you don't have to compromise the dose of one of the drugs. In here, you do a little bit, and so whether or not it's the right strategy, I think we just need to sort that out based on larger studies.
Yeah, I would just add it was a close to 70% response rate. What we don't have is a lot of correlative data from that. One of these agents targets Nectin-4, one targets TROP2. Are they both expressed on the same cell? Are they expressed on different populations of cells in the microenvironment, which makes it even more attractive? One of the thoughts I had actually after seeing the data was, giving them together also has an increased risk for toxicity, which is expected for both of these agents. Could you do an approach where you gave one agent and then the next cycle gave the other agent, sort of an intermittent approach? So you can think of all these perturbations of what to do, and you obviously might compromise efficacy, so I don't think that's the first study I would run to. But if you're really trying to get at sort of mechanisms of resistance, maybe these are different ways to sort of address that. And I completely agree that adding a checkpoint inhibitor here and moving this up in the treatment regimen, at least for pretty fit patients, might be fruitful. We'll have to see.